Right, good afternoon. So today we're going to take a look at the 4 levels of protein structure. And so there's 4 levels. There's primary structure, secondary, tertiary, and the last level is quaternary structure. So what are they? Every biochemistry student should be familiar with this. The first level of protein structure is going to be the linear sequence of amino acids. That's called primary structure. And so in Mo, I can simply display the primary structure of a protein. If I go to my protein builder, all I really have to do is just start with, here's the 20 amino acids. Let's just start with glycine. And I'm just going to call this primary, for example. First of all, to start off your protein, I'm going to go ahead and show atoms and just make them a little more visible. This is the amino acid glycine. It's the simplest amino acid. At the center of every amino acid is an alpha carbon, and then it's an amino acid. So there's an amino side, and there's a carboxylic acid on the other side. Now, one thing you will notice is this amine is a quaternary amine, so this actually has a +1 charge. And the carboxylic acid is missing its hydrogen, and so this is actually in base form, right? Because this is modeled at physiological pH. The pKa of the carboxylic acid side is approximately 2. The pKa of the amine side is approximately 9. And because we're at a pH of 7, that means that the amine should be fully protonated. It has not been deprotonated yet until the pH rises to at least 9. And the carboxylic acid is deprotonated because we're at pH 7. We've lost our proton long ago because this is a fairly acidic residue. And then down here on the bottom we have a hydrogen. And every amino acid has one R group. In this case of glycine, we have a hydrogen as its R group, right? Now, if I add another protein together, let's just add histidine here. Here's a histidine residue. Notice here's my glycine, here's my histidine side, here's my alpha carbon right here. And I still have an amine and a carboxylic acid. The carboxylic acid is open over on this side. The amine is actually bound to the carboxylic acid of glycine, and so that's actually forming. This is a carbonyl with an amine next to it. That's actually called an amide functional group. So the amide has partial double bond character, and it is somewhat planar. So that's a rigid bond. Right? Now it's also important to point out there's 2 angles on either side of both of these alpha carbons right here, right? So the amine to alpha carbon, that's called the phi angle, and the alpha carbon to the carboxylic acid is called the psi angle. So there's also in histidine, there's also a phi and a psi bond angle as well, right? So we've added, this is a dipeptide, I can make a tripeptide, I can keep going, and we can make them as long as we want, and so that is just the primary structure of this protein chain, right? And we can look at the primary structure in our sequence editor, and we can display that with a 1-letter code, or a 3-letter code right there like that, right? So that's primary structure. Now, this is obviously the protein is not going to look like that. We need to adopt different conformations. We have those phi and psi bond angles. So let's try and let's try and fold. Once the protein is synthesized at the ribosome, well, what's going to happen is we're going to start to fold hydrogen bonding interactions, and let's see. So if I now let's just start with the glycine. And I'm going to go into an alpha helical arrangement. So alanine, cysteine, isoleucine. Let's just put a few together. All right, I think that's enough to illustrate what we're trying to do here. Now, what I want to do is put the ribbons on so we can see a coil structure, right? And with this structure, this is called an alpha helix, and it's one element of secondary structure. So the amino acids are going in order from the amine to the carboxy terminus. Here's the amine terminus. Here's the carboxy terminus. And as they folded up, they've started to form. You can actually see these little blue arrangements. These are hydrogen bonding interactions between the carbonyl and the amine. If I undisplay the ribbon temporarily, you can actually kind of see that the hydrogen bonds are between the carbonyl right here and the NH. And so that's consistent throughout the protein, right? So, and alpha helices are very stable. We'll also know that as we go through one complete turn of the alpha helix, it's approximately 3.6 amino acid residues. So there's 1, 2, 3, and then we're getting to 4. 3.6 is close to 4, so we've actually went through one complete turn of the alpha helix when we get to all 4 of these residues, right? And then the rise per rung, the rise per residue is about 1.6 angstroms. And so if I kind of, let's see if I can measure the alpha carbon here. Let's see if I can find another alpha carbon. So we got about 5.4 angstroms. Now that's 5 angstroms going through one turn. The rise per residue, right? If I kind of, let's turn this off. The rise per residue, that's just like one residue. That one residue goes through only about 1.6 angstroms right there, right? And another residue is another 1.6. So we can we've got some very specific measurements we can measure. So that's an alpha helix. The other secondary structural arrangement is what we call a beta sheet. And I have an example of a beta sheet right here. In Mo, they color these in yellow so we can see those. And what we see also, there's 2 types of beta sheets. This is actually an anti-parallel because it kind of alternates. It goes one direction and it goes the other direction and it goes the other direction. So this is an anti-parallel beta sheet. A parallel beta sheet would have the amino acids going all the same direction. The other thing we see is that we do have some hydrogen bonding interactions, and we can actually see here's one right here in the middle between the carbonyl and the amine, the NH right there. There's another one over here, right? And so we can measure, let's see here. It's a little hard to measure there. I think I might have to turn off my ribbon. Oops. Want to turn a little bit more off. Now we should be able to see. And we're going to measure heavy atom to heavy atom, and there's a double hydrogen bond. There's another one, and so we can see multiple hydrogen bonds. And they're about 3 angstroms, take or give a few there. And so when we look at a beta sheet, they are stabilized by hydrogen bonding interactions as well, right? Now once we start to fold, this just starts to form. That's why we're only at secondary structure. right? The next level is actually tertiary structure, and this is this kind of is mediated by energy landscape theory. So we kind of fall down a funnel-shaped energy diagram, where at the top of the funnel we have high entropy and things are kind of unwinded. As we fall down, we finally arrive at the native state. And so this is the folded conformation of the protein. And this is a glucose-galactose binding receptor, which is in E. coli. You can actually see the ligand in the middle here. And we'll go ahead and put that in space-filling form. So there's the sugar. And so the conformation of this is picked up by another protein system, which enables E. coli to take in glucose, right? And so, again, every protein has this 3D shape. Now, if we look at everything, if we look at all the atoms, this is kind of what you would see, well, minus the colors, which are CPK colors. This would be the kind of the shape you would see in the amino acid. And actually, probably a better way to display that would be with a molecular surface, right? So if I go to surfaces and maps, I can actually display a molecular surface, 2GBP, and I'm going to say selected atoms, and we can color this in a number of ways. Let's just color it by electrostatics with negative charges in red and positive in blue, and create our surface. So if we were to look at 2GBP, this glucose-galactose binding protein under a microscope, this is more or less what you would see. Now you wouldn't see the colors, but this is the shape that you would see, right? So that's kind of what we see with, that's tertiary structure. And let's go ahead and delete that. The other thing we see is the whole thing is basically one long ribbon, right? That's one chain. And so the amine terminus is alanine right here, and that's selected. That's down here at the bottom, and then it goes through all these conformations, and the carboxy terminus is at the end. That's lysine. Here's the carboxy terminus out at this end right there, right? And so that's all one chain wrapped up like a plate of spaghetti, right? So that's tertiary structure. Then the next level is quaternary structure. And so that's essentially two or more chains interacted together, right? And in this case, we're going to hide the atoms, but this protein is insulin, and this is actually a hexamer. So we got two chains on one side, two chains up here, and then two chains down here. And so this is insulin, and this is present in your system. It's produced by the beta cells of Langerhans in the pancreas, and this is very important for binding to insulin receptors in the liver and the muscle cells and systemic cells that causes those cells to take up glucose in response to high levels of blood glucose, right? So this is very important, and it's a quaternary level of protein structure because there are 6 chains interacting in a hexamer arrangement. So that's where we've got all 6 of those chains all interacting via 9 covalent interactions together, right? So I hope you've enjoyed this video. I hope this was useful, and thank you. You can leave a comment if you find this useful. You can subscribe. You can like to the channel as well. So thank you.